Chuck.
Really appreciate it.
Wow. I wish my children were here. God. Maybe we have a videotape I could send them on YouTube. Well, so I guess, I'm at the U. That's what we call the University of Miami. And you're at the U, and you're at UM, and I'm at UM. And I've had a very close relationship with this department for many years. For those of you who are as old as I am, which is almost no one, I was the consultant to the dean when there was a search for a psychiatry chair. And so you can blame me in part for Dr. Schultz, who's done a phenomenal job as chair here. And I just want to say a few words about him before I lecture to you and then try to run off and catch my plane. And I apologize for not being able to stay longer. But I have a birthday this week, and my wife is taking me on a trip tomorrow, and I want to stay out of the doghouse. So, these jobs are not easy. When I started as a chair about 26 years ago, the jobs, they were a lot of fun. There was money coming in and clinical income. You could use it for research. There was philanthropy coming in. NIH funding was relatively bountiful, although we didn't say so at the time. And, the great thing about being a chair was that you could make people happy. And the world has sort of changed, and it's tough now. And, if you haven't noticed, we're not neurosurgery or cardiac surgery, and we don't garner tremendous clinical revenues. And for that reason, it makes our job tougher. And I had the pleasure of having breakfast with the dean this morning, along with Dr. Schultz, and did everything I could to try to convince the dean, who didn't need much convincing, actually, about how important mental health is and psychiatry is, and about the people that we serve. And I, often think that I have the best job in the world, and all of you do too, because you're in the medical specialty, which allows you to hear people's life stories. And what an amazing privilege that is. And every time I see a new patient and I hear a life story, I think, thank you, God, I love my job. And I hope all of you are as excited about it as I am. So the next time you see Dr. Schultz walking down the hallway by himself, walk up to him and give him a hug, okay? Because every once in a while the chair needs a hug, because it's tough out there. He's out there battling to protect you against forces of evil, okay? So let me try to tell you a story. And the story is about where I think the future of psychiatry is going. And Chuck specifically asked me to talk about this topic. I want to provide you with my financial disclosures, which are listed here. They're also on the University of Miami website. And if you have any questions about them, anybody wants to buy any of my patents, I'd be more than happy to sell them to you because they're worthless. But, just in case you want to take a gamble, let me know. So I thought I was going to give this talk in February in Minnesota, and I thought I'd include this slide of Key Biscayne, which is about 10 minutes from my office. But, I would never recruit anybody from Chuck's department because we have honor in not trying to ever harm each other and trying to help each other be successful in this sometimes tough world. So I think the fundamental unanswered question for psychiatry at the current time has to do with why our patients develop the disorders that they do. No one ever says, when they're in high school and you interview kids and say, what do you want to do when you grow up? No one ever says, oh, I want to be a heroin addict. Or, you know what, I'm going to be a schizophrenic. Or I'm going to have bipolar disorder. So, contrary to what people might think in the lay public, our patients don't volunteer to become ill. And until we fundamentally understand the pathophysiology of these illnesses, we're never going to fundamentally understand what the best treatments are for these people. And what personalized medicine fundamentally is it asks two questions. One, what are the most significant factors that contribute to vulnerability to develop an illness. And whether you're talking about breast cancer or ovarian cancer or cystic fibrosis or ALS, and don't dump an ice bucket on me now. Brilliant strategy, I might add. Or whether you're talking about bipolar disorder or suicide, you know, what is it? What combination of factors result in people developing these disorders? And the rest of medicine, as you'll see, has been a bit more advanced than we are, but we have to get there if we're going to advance the field. So the fundamental question is, what causes someone to have post-traumatic stress disorder or major depression or bipolar disorder? And fundamentally, what it's about is about genetic risk, and I'm going to talk to you a lot about that, because unlike many other disorders in medicine, our disorders are by and large genetically driven. And guess what? We don't pick our parents. So our patients, they get what they get in terms of inheritance. And a lot of folks are starting off with a tremendous vulnerability to develop a psychiatric illness. And then, of course, there is a tremendous environmental impact. I spent most of my career studying child abuse and neglect and how child abuse and neglect interacts with genetic vulnerability to lower one's threshold to develop these devastating disorders. And the question is, how do genes and the environment interact to result in changes in cells, particularly during development, because we know that development is critical here. And remember, the human brain develops very slowly. So the adult brain isn't clear until about age 24. Well, those of you who have teenagers know that, right? They're not very mature, and they sometimes make bad decisions. So how do these cellular changes result in brain changes, circuit changes, that end up increasing the likelihood of developing a disorder? And we could be talking about schizophrenia, we could talk about bipolar disorder, we could talk about major depression, and I'm going to talk about largely mood disorders and PTSD as examples of the kind of work that I think will yield some fruit here. So this is an old slide. Just think about bipolar disorder. I pick it because 2/3 of the risk to develop it is genetic. Okay, 2/3. There aren't that many diseases in medicine other than single gene mutations that have such a high genetic load. And so the question is, what is it about some combination of susceptibility genes and protective genes in combination with environmental factors that result in this pattern? And all of you know the classic pattern of bipolar disorder is early onset depressions, often prepubertally, followed by that first manic episode, and then a series of episodes of mania and depression. What is it biologically that occurs? Is it epigenetics? Is it changes in gene expression? What is it that occurs? And within the panoply of what we categorically call bipolar disorder or major depression, are these one disorder? Are they 10 disorders? Or are they 100 disorders? And this debate is going on in all of medicine. You don't have to feel like somehow we're disadvantaged in psychiatry. I went to a meeting that Chuck went to last year, the year before, on breast cancer. And I listened to all these experts on breast cancer debating about the risk factors for breast cancer: obesity, family history, exposure to various hormones. And I finally listened to all of this debate, and I raised my hand gingerly and said, well, just tell me, is there one type of breast cancer? Are there 5 types? Are there 10 types? Are there 200 types? And all hell broke loose in the room, because they have just as much of a disagreement about this than we do. And interestingly enough, and you all may know this, and I didn't know this until I went to this meeting, is that breast cancer changes in a given individual. So the breast cancer you're originally diagnosed with actually mutates and changes in the course of the illness, make it even more complicated and difficult to treat. However, when I was a medical student, the 5-year survival rate for breast cancer was about 20%, and now it's 91%. And the reason is personalized medicine. Because what breast cancer oncologists have been able to do is to come up with genetic markers that predict treatment response. Estrogen receptor positive or negative, BRCA1, BRCA2, these are Herceptin. These are important characteristics of the tumors that predict treatment response. And that's what we're lacking in psychiatry. So where personalized medicine is going to take us in psychiatry is away from here. So now patients come to us and they've had a suicide attempt, they're having an auditory hallucination, they're morbidly depressed, and we see them, or they have cognitive dysfunction. And notice that when we see them, we treat them, but there has been some relative impairment of organ integrity. They're jaundiced, they've been drinking for 30 years, they have cirrhosis of the liver, their brain's been marinated. That's probably not reversible. And where personalized medicine will help us is being able to identify patients before the tipping point. What are we doing in Alzheimer's disease? We're focusing on MCI, pre-MCI, patients who don't yet have a mini mental state score of 12, but have a mini mental state score of 26. And maybe we could prevent the progression. We need to do that in schizophrenia, in bipolar disorder, and in major depression, and in PTSD. And here's where we want to be. We want to be able to identify who is vulnerable for these diseases so we could develop an at-risk group. And we're in the midst of a big NIH study looking at post-traumatic stress disorder. Only 35% or approximately of patients exposed to the worst trauma develop PTSD. 65% do not. We know that from 9/11. And if you try to treat everybody, people will develop it who wouldn't have developed it if you force them to get treatment. That's why we have defenses. And what this grant I have is to try to figure out what are the predictors of developing PTSD. So let's talk about genetics. All of you have seen these cover stories. They're pretty impressive, right? The future of medicine, the secret of life. We sequenced the human genome. It told us everything we wanted to know, right? Not right. Not right. You know, Jim Watson, who discovered the double helix, when this was accomplished, he said, we used to think our fate was in our stars. Now we know in large measure our fate is in our genes, paraphrasing Shakespeare. Well, maybe it isn't, okay? Maybe we don't understand the power of the human genome yet. But the fact is that psychiatric disorders have the highest of all the complex disorders, hypertension, heart disease, cancer. Psychiatric disorders have the highest genetic valence. The largest contribution to vulnerability is bipolar disorder, 2/3, followed by schizophrenia, 50%, followed by PTSD, about 40%, followed by depression, about 35%. Huge genetic contribution. This is an old study that I like to use because it's a summary of a number of studies which should convince you that bipolar disorder is fundamentally a genetic disease. This is the concordance rate between dizygotic and monozygotic twins. If one twin has bipolar disorder, what's the likelihood the other will? And in every study, the concordance rate for monozygotic twins far exceeds that of dizygotic twins, even if separated at birth. So clearly this is a genetic disorder. For major depression, it's about 35% risk for major depression is genetic. But the problem with our current nomenclature is that we group together a large number of what are clearly disparate biological entities into a single diagnosis. Each of these mood disorders can in fact fulfill criteria for major depression. And I think all of you would agree that an 80-year-old who's never been depressed in their life, who's got a brain full of white matter hyperintensities, who has a first episode of major depression at age 82, is biologically very different than a woman who is suffering postpartum depression or an individual who had their first episode of depression as a youngster. These are fundamentally different illnesses, but we categorize them as the same. It would be like studying everybody with hypertension, where somebody had a pheochromocytoma and someone else had renovascular hypertension and someone else had yet another cause of hypertension, and grouping them together and trying to understand the disease. These are different entities. So before we sequenced the human genome, we used to do linkage studies, and we would try to find places on the chromosome which seemed to carry risk for mood disorders, largely bipolar disorder, and a number of these locations were identified on various chromosomes. And I'm going to get back to that in a minute. In the meantime, Chuck and I, using the model that, you know, if you lose your wallet in the park, you should look under the street lamp, even though you lost it a quarter of a mile away. All we had at our disposal were neurotransmitters, and our favorite neurotransmitters was serotonin, norepinephrine, and dopamine. In fact, serotonin was so much my favorite neurotransmitter that my daughter, who is now applying to law school, thank you God, whose name is Sarah Francis, said to me one day after hearing me give a talk like this, sitting in the back of the room with her coloring book, on the way home in the car, she was very quiet, and I said, "Sarah Francis, what's the matter?" And she said, "Daddy, who's this little girl serotonin you keep talking about?" So I told her she would be on an analyst's couch one day having some rivalry with the neurotransmitter. So what about these neurotransmitters? Well, the problem is, as much as we know about them being involved in the depression, in this day and age, you have a patient in front of you who's depressed, and you have no clue whether they have a norepinephrine problem, a serotonin problem, or a dopamine problem. And I'll show you a slide that suggests each of them may be involved in depression, but the sad fact is, when you're picking a treatment for a given patient, talking now about antidepressants, not psychotherapy yet, you don't know what's wrong with them. And all of you think you do. I mean, I do. I think I'm a pretty good psychopharmacologist. I'm sitting there, I see a patient, I say to myself, oh, this is a venlafaxine responder, I could tell. Or, you know, this is an S-citalopram responder. Well, all of the studies that we have showed, no, we don't. You know, not at all. Oh, this patient is having trouble with sleep, I'll give them a tricyclic. No, doesn't work that way. We have no good predictors, really, and I'll talk about that near the end of my presentation. So the trouble is that depression is a really complicated illness. It affects a whole bunch of different brain areas. We know that from brain imaging studies. So the notion that a single neurotransmitter is responsible for depression is pretty silly. I mean, we're talking about, think about it, 100 billion neurons that make 800 trillion connections. And you're telling me that serotonin is responsible for depression? I don't think so. Not if it affects every one of these brain areas that are involved in the manifold symptoms of depression. So here's serotonin. Here's one of the gazillion serotonin nerve terminals. There's the serotonin transporter. You know what that is. That's the protein God created so you could prescribe SSRIs. So there it is, and you prescribe SSRIs, and it blocks the transporter, and serotonin hangs out here and stimulates postsynaptic receptors. That's great. But you can use it as a marker for serotonin neuron integrity. And you could do studies like we did here, in which you measure serotonin transporter binding in drug-free depressed patients, some who've never been treated, versus controls, and you see some reduction in the RAFE, suggesting maybe there are, as the postmortem study said, fewer serotonin neurons, for example. But let me give you a news flash. I just finished a large center grant study of never treated depressed patients. I'm going to present this at ACNP, where we randomized never treated patients to duloxetine, escitalopram, or CBT. And the question is, does serotonin transporter occupancy as measured by PET, does it predict outcome? Number one, no, it doesn't predict outcome to one or another of those treatments. Number two, does how much the drug occupies of the transporter predict outcome? So you would think if you occupy 30% of the serotonin transporter, maybe you get less of an effect than if you occupy 90%, right? Wrong. There was no effect whatsoever on either serotonin or norepinephrine transporter occupancy in predicting response to either escitalopram or duloxetine, or for that matter, CBT. So it surely is not all about serotonin. What about dopamine? Well, I mean, dopamine, if you had to pick a transmitter that's involved in the depression, it should be dopamine, right? Because it's the neurotransmitter that mediates pleasure, right? And if there's one pathognomonic sign of depression, it's the inability to experience pleasure. So you would think it's got to be about dopamine. I mean, whether it's food, sex, drugs, rock and roll, it's all about dopamine. And there is some evidence from imaging studies that dopamine transporter binding as a measure of dopamine neuronal integrity is reduced. These are depressed patients by age. These are controls. They are reduced. But the problem is, look at how much dopamine you lose over time as you age. That's why there aren't any 60-year-old tennis pros, right? It's not your fault. You just can't swing the damn racket anymore. Okay. And then norepinephrine, the first neurotransmitter thought to be involved in depression. And there is evidence from postmortem studies that tyrosine hydroxylase is elevated in depressed suicide victims in an attempt to make more norepinephrine. But again, neither norepinephrine nor dopamine seem to be fundamentally involved. And you know the treatment data, be very clear about it. Only 28% of patients in the STAR-D trial got into remission on up to 40 milligrams of citalopram. 28%. If you're an oncologist and you said to somebody, on average, you're going to get a 28% reduction in your tumor load, or you'll be tumor-free, but 72% chance you won't, you wouldn't be very happy. So our treatments are not as good as we thought they were. And even as you went down the steps in the STAR-D study, patients did respond to increases in switching and combination, but there were very high rates of relapse. So we're not as good at this as we thought. So what about genetics? Let me surprise you a little. All of you know what this model is. You learned it in high school. You learned it again in college. You learned it again in medical school. DNA is transcribed to RNA. RNA leaves the nucleus, goes to the ribosome in the cytoplasm, and you transcribe the protein, right? And here's the human genome. I wanted you to see it because it's so insightful. There it is. The small portion of it. It's the triplet code. Are you not excited? Look at that. It's really cool. Now, the reason that we don't all look alike, that we're not clones of each other, is because there are little variations in the human genome called single nucleotide polymorphisms. You're all aware of that. There are about 5 to 8 million of them. And that results in a great deal of genetic variability. Now, some of those variations have no particular biological impact. Here's an example of a substitution in this very first protein. This is an alanine amino acid, and you convert an A to a G by a spontaneous mutation, but it still codes this triplet code for alanine. The protein remains the same. But sometimes these SNPs are in fact functional. So in this case, a G goes to an A, and now you code instead of for arginine for a lysine, and that changes the tertiary configuration of the protein. So now it's a better receptor, a worse receptor, a better transporter, a worse transporter, a better enzyme, a weaker skeletal protein. It has a functional consequence. Who cares? Well, we care because it turns out that some of these functional SNPs actually confer vulnerability or resilience to our diseases. And this study, which was published in Science, demonstrated the importance of a functional polymorphism in the serotonin transporter. And there are two forms of this, two major forms. There are other forms, the long arm and the short arm. And you see the importance here is that the short arm actually has reduced activity. So it's functional. And this paper was published in Science now 10 years ago, and essentially turned our field on its head. I'll just go over this with you for a minute. What it essentially showed was that depending on the genotype of the serotonin transporter, if you were exposed to child abuse, you were either totally resilient to depression, you didn't develop it, or depending on if you were a heterozygote or a homozygote, you were remarkably vulnerable to it. The critical finding here is not only that the serotonin transporter polymorphism predicted vulnerability to depression in the presence of severe child abuse in this New Zealand population, but in the absence of child abuse, it didn't matter what your genotype was. Only in the presence of this environmental stressor did the disease vulnerability become clear. And that opened our eyes to the fact of gene-environment interactions. So for a long time, I've been interested in stress and the role of stress in the biology of depression. We know that CRF or CRH is the brain's neuropeptide neurotransmitter that controls stress, as you all know. And I won't go over all the details that this neurotransmitter is hyperactive in depression and in PTSD. But I want to give you an example of the kind of studies we could do. So we studied about 500 patients, largely African American and inner city. This was a highly indigent population, you could see from their income. They were all waiting to be seen by an internist at the Safety Net Hospital. And we obtained Beck Depression Inventory scores on them, as well as measured child abuse by the Childhood Trauma Questionnaire. And the more child abuse they had, not surprisingly, the more depression they had. And then we divided the patients up into moderate to severe child abuse and none to mild child abuse. Those are their Beck scores. And then I just want to show you the kind of work you could do. So if you think CRF is an important neurotransmitter in the biology of depression, you could take the CRF receptor polymorphisms and you do the same kind of studies that were done with the serotonin transporter. And what you find are three SNPs. One is a protective SNP. No matter how much child abuse you get, if you have this SNP, you don't get depressed. And then you have three of the opposite vulnerability SNPs. If you have the bad luck of having this SNP, you're going to get very depressed. These are Beck scores in the presence of child abuse. And again, look, there's no effect of the genotype in the absence of this early life trauma. What if you have 2 or 3 of those vulnerability SNPs? Well, then you have a very, very high likelihood of getting depressed in the face of child abuse. What if you have all the protective SNPs? Well, then no matter what happens to you, you're not going to get depressed. And we've gone on to show that this is also true with other genes for post-traumatic stress disorder, that a gene that controls the signal transduction of glucocorticoids called FKBP5, those SNPs mediate the likelihood of you developing PTSD. This is done in that same population. And that adult trauma and childhood trauma are additive to the PTSD vulnerability in folks who have these genotypes. Now, lastly, we just published this in Nature Neuroscience. We've been finally able to uncover the molecular mechanism by the way this works. And it turns out there is an epigenetic effect associated with child abuse that acts on FKBP5 and only that vulnerability allele in order to underlie the vulnerability for PTSD. This means that we can develop targets, molecular targets like this, to administer to patients to try to prevent them from the sequelae of PTSD after trauma. So this is a big effect. This is looking at childhood trauma questionnaire scores, and you see this tremendous effect here in those who have the FKBP5 risk allele in terms of this epigenetic methylation. You don't see it in the FKBP5 protective allele. So I'm going to skip, for lack of time, a couple of things because I do want to talk about prediction of treatment response. So it turns out there are about a dozen genes that we and others have now isolated whose variations predict whether you're going to develop either major depression or PTSD. This is a particularly interesting gene, and I'm showing it to you for several reasons. One, my name is not on this paper. My former resident's name is the first author. My former faculty, my current faculty at Miami, Amanda Myers, this was published in Nature, identifying a gene called the pituitary adenylate cyclase activating peptide. Its receptor, variations in its receptor right here appear to determine whether women are vulnerable to PTSD but not men. This is the first finding that finally seems to explain why women are so much more vulnerable to PTSD than men. And we did this in not only an original sample but in a replication sample of subjects. So the total number of subjects we looked at was more than 1,200, and this effect only occurs in women, as you see here, both in the original sample, and it turns out this gene has an estrogen receptive element. And more recently, my vice chair for research, Klaus Wellstedt, who Chuck had a chance to meet, with my former PhD student who's just been named the director of the Max Planck Institute for Psychiatry in Munich, the first woman ever named to be the director of a Max Planck Institute. I'm very, very proud of her. And my former resident have identified yet another gene, an opioid receptor-like protein gene variant that appears to also play a role in mediating PTSD. Now, it turns out then that we're going to have a panoply of about 10 or 12 genes on our radar screen, and we're going to get very shortly human genome wide scanning on every one of your patients. And they're going to be part of the medical record, and you're going to be able to look up and determine how many of these vulnerability genes do they have. Currently, it's an academic exercise because we don't know how it affects treatment, but it'll allow us to do more homogeneous studies. In the meantime, those old association studies, linkage studies that I showed you, have given way to GWAS studies, and now we're beginning to learn more about mood disorders by virtue of these very large GWAS studies. And so this paper was published this year, a very large study, as you can see, on bipolar disorder, and it identified a number of genes. This was a sample of 24,000 patients, and a number of genes were identified far beyond the chance. And these genes relate to calcium channels, which open up an entire new avenue of research for molecular targets to treat this devastating illness. This is where the future of psychiatry pharmacology is certainly going to go. And what's nice about these findings, these two genes, is they were replicated in samples from all over Europe and Canada. And as you can see, it's rare to get this kind of uniformity of a finding in studies of psychiatric genetics. So I'm pretty excited. So now let me really surprise you. So how many genes are there in the human genome? It turns out there are 22,000. Now, if you look at genetic books before the human genome was sequenced, the estimate was we were going to have 100,000 genes. And that's an example of human hubris. We thought we're so sophisticated, we're going to have the most genes. But you know what? Look at this is number of genes as a function of species. Okay? So check this out. We have slightly more genes than C. elegans, the sea worm, a little bit more than the fruit fly, but we have fewer genes than seaweed. Okay? So if it's not, like, what's that about? What then programs our complexity if it's not the number of genes? What have we learned from the Human Genome Project? And it turns out there's a shocking finding. Is what's really impressive about us as a species is not DNA, actually. It's RNA. Well, you'll say, well, that doesn't make any sense. DNA codes for RNA. So this is what we were all brought up with, and if you're on Jeopardy, you're going to have to give this as the answer, okay? So yes, DNA is transcribed to messenger RNA and it makes protein. Except it turns out that the vast majority of DNA does not code for messenger RNA that makes protein. The vast majority of DNA was originally called junk DNA because it didn't code for protein. Well, now, do you think God made this much DNA and it was going to have no function? So, like, let's plot the amount of non-coding DNA as a function of complexity of a species. And now look. Oh, prokaryotes, simple eukaryotes, there's amoeba, complex fungi, whatever that is. And here we are at the very top. Wow. So the more non-coding DNA you have, the more complex you are. Well, how much of our genome actually codes for protein? Shocker. 98.8% of our genome doesn't code for protein. It's not that it's not important. It codes for all of the enzymes and receptors and transporters that you're interested in. So what is going on? It turns out that the new view of genomics is that there are islands of conventional protein coding genes, but they're completely regulated by RNAs, microRNAs. And these microRNAs control transcription and translation. And we're already seeing publications about microRNAs being unique in schizophrenia and in other psychiatric illnesses. So in the last few minutes, I'd like to talk to you about this road that you're on every day. This is the mood stabilizer highway. And you're sitting out there and you're trying to decide for your patient, should I prescribe lamotrigine or lithium or CBT? What's the data? What about antidepressants? Am I going to be yelled at by my supervisor if I prescribe antidepressants? No, you shouldn't be. Okay. Or you're on the antidepressant highway and you're trying to figure out, should I prescribe this new antidepressant, vortioxetine, or should I prescribe the cheapest one that's on the $4 Walgreens list? What's best for my patient? If it was my sister, assuming you like your sister, what would you prescribe for her? And so in 2004, I published this paper in the American Journal of Psychiatry, which is the least cited paper I've ever published. So I'd like to think it was mildly before its time, but it asks the question, there's an antidepressant named milnacipran that's available in the U.S. to treat fibromyalgia and levomilnacipran is now available as well. But at the time, it was only available in Japan and Spain. It's a SNRI. And I wanted to look at whether polymorphisms in the norepinephrine transporter would predict treatment response. And we discovered that there was a SNP that predicted a poorer response to milnacipran, a SNP of the norepinephrine transporter, and another one that predicted a slow response. Two different norepinephrine transporter SNPs. We were interested in the CRF system, and we published this report in the archives where we looked at the STAR-D data. We were able to come up with some associations of polymorphisms in the CRF system. And then Elizabeth Binder did this phenomenal study looking at FKBP5, this glucocorticoid chaperone protein, and whether variations in it predicted response. And I want you to just see this data published in Nature Genetics, largely ignored 10 years ago. But look at what a profound effect this is. So let's look at the tricyclic data. So if you have this SNP, you have a great response, and if you have either of these other two, you don't. The same with Remeron, the same with tricyclics. So wouldn't you want to know this? If you had this data in front of you and your patient had one of these SNPs, wouldn't you want to, like, say maybe psychotherapy is the way to go? Maybe let's go with CBT first because you have less of a likelihood. And if you knew this for MAOIs and other antidepressants, it could be helpful. So in the last few minutes, I want to talk to you a little bit about an exciting new area, which is inflammation. There's a lot of evidence that inflammation in the form of pro-inflammatory cytokines may be playing a role in certain forms of depression. And the data is remarkably in agreement here. But the bottom line is, not only because we were one of the first to report it, but that an unusually large number of depressed patients, particularly refractory patients, in fact, show elevations in inflammatory markers, particularly interleukin-6 and C-reactive protein. And so one of my colleagues, Andy Miller, did this study looking at a tumor necrosis factor alpha inhibitor. And I just want to show it to you, these data, as an object lesson about what you could learn about clinical trials. So he gave this drug, infliximab, which is a TNF alpha antagonist, to treatment refractory to patients, 30 per group, or placebo. And he gave the infusion 3 times at baseline, week 2 and week 6. And trust me, there were patients by history who failed at least 2 trials, and they had major depression by SCID, and they were exactly the kind of patients you'd think they were. 2/3 were women, and 40% were unemployed because of their depression, a pretty highly educated group, and they were very depressed. And Andy called me, he was in tears. The study failed. Here's the results. Study failed. Infliximab versus placebo. If anything, placebo looked a little better at the end of 12 weeks. Terrible. Really depressing. Andy's Hamilton score was about 18. I said, Andy, wait a minute. Why don't you look at their baseline inflammation and see whether that has any effect? And he's like, wow, look at this. So yes, the drug failed. If anything, placebo looked better. But look what happens as the evidence of inflammation increases. The patients with CRP levels greater than 5 had a great response. Those above 3 had a good response, and these didn't. The point being that depression is not a homogeneous entity. And when he divided up the patients to those who had elevated CRP and those who didn't, well, it turned out that the patients got worse with the drug if they didn't have inflammation. Inflammation is probably, you have to have a sweet spot. I mean, we have inflammation for a reason, right? It's evolutionary evolutionarily important. So we're going to have to begin characterizing our patients as having inflammation, and maybe then we're going to use certain treatments for them or not, and we're going to use others. You have to hit the sweet spot. Until we understand the pathophysiology of our disorders, using this as an example, then we're not going to be able to do much more than trial and error. So I like this quote from Steve Jobs, who quoted Wayne Gretzky. He said, "I skate to where the puck's going to be, not to where it's been." We've got to get away from SSRIs, SNRIs, 5-HT2D2 antagonists. We need to begin to understand what the underlying pathophysiology is to our disorders, so then we can embrace personalized medicine. And I think I'm out of time, so thank you very much.